Hello, and welcome to a quick tutorial on how to use the University of Scranton's UV-Vis spectrometer in the chemistry lab. To begin with, you will have to ensure that the instrument is on. There's a on-off switch on the side, and the single line is the on part of the switch. Next, go to the computer and you will see a gray eye icon. That is the UV-Vis software. Next is taking your measurements. To do so, put your sample inside of a cuvette and wipe it thoroughly with a Kim wipe to ensure that there are no fingerprints that may interfere with your results. This particular cuvette that was used in this video is four-sided, meaning you can put it in any direction that you would like. However, some cuvettes are different, so if you have any questions, you can always ask your professor. Now, the first measurement will be performed on a blank sample, meaning it will only be performed on the solvent that you used and nothing else. This is so none of the results can be attributed to any sort of background noise. Once it's loaded, there is a button in the toolbar on the top that looks like a red line with two red arrows over it. Click on it, and it will tell you to put your sample into the cell, in which case you already have, and you can hit proceed or just click enter on the keyboard. If you ever wonder how long you have until your measurement is up, there's a progress bar in the bottom right corner of the screen. If you're measuring specific wavelengths, time is usually not much of a concern. However, if you're taking a full spectrum, like in this example, then that's a good gauge of how much time you have left. Once that's all done, you are now free to begin scanning your samples. Be sure to remember to wipe each one thoroughly with a Kim wipe. You can't afford to forget this step because otherwise, your numbers may not be exactly accurate. For this reason, it's also recommended that you hold the cuvette itself by the top, because the light that shoots through in the UV-Vis instrument will not interfere with that part. Be sure to close the instrument once it is placed inside, and then you may start. In the same toolbar that the baseline button is located, there's also a red running person button. That is to run the samples. So click on that, then it will ask you for the name of your sample, which you can rename to anything distinctive that'll help you remember which one is which, and then hit OK and proceed. Now, as you may have noticed, the data was peaking at around 2 points. Relatively speaking, this is a dilute solution of green food coloring. And as this data shows, there appears to be 2 small peaks around where yellow and blue absorb light, which makes sense in color theory since yellow and blue combine to make green. Using these numbers, you have to approximate it, unfortunately, because finding the Y value is unfortunately quite difficult. You can find the ratio of the yellow dye to blue dye that was used in the manufacturing of this, which is the importance of using an instrument such as this. However, to get these exact numbers, what you can do is then switch to a fixed wavelength experiment, which we will do now. Using this graph icon up here, we can actually change our method and approximate the wavelengths that our 2 peaks were at. So for this example, we're going to use these two numbers, and we input them manually into the 1 and 2 slot of this method editor. Now, if you change your method at all, one thing you will have to do is blank again. So that's what we're going to do here. Since you know the process by now, I'm just going to fast forward through it, but know that you're going to have to do this each time you edit your methods whenever using the UV Viz. But otherwise, you can load another sample straight after loading another if your method is the exact same. Alright, so now we have Kim wiped our sample once again and loaded it. And this method will be much shorter because it's only scanning 2 wavelengths instead of scanning across an entire range. Now a table will show up that has numbers, and these are the exact absorbance values that the UV Viz spectrometer has interpreted. So the ratio between those two components that I was talking about earlier, you can do those with this method. Once you're finished, unload your samples and dispose of them as necessary, and then you can simply X out of the software. It will ask, do you want to save the changes to your method, in which you don't have to. In fact, it'd be more trouble if you did. So answer no. And then the final thing to do is log off the computer and ensure that the instrument is turned off, which means that the circle is pressed.